let's keep going. So we finished the most difficult part. And that's the example we talk about the sodium glucose importer. So they use the concentration gradient of sodium as the energy source. They usually pair with the primary pump. And this pump creates the concentration gradient of sodium. And this sodium glucose importer use the concentration of gradient of sodium to move glucose against the concentration gradient. And in the cell's point of view, we always look at the cell's point of view. You still need to use ATP to make this whole process to happen. So we call this secondary active transport, also called indirect active transport. And the molecule can move in the same direction, like the sodium glucose importer. That's the example. They move in the same direction. And they can move in the opposite direction. We call it antipolar, like the sodium hydrogen antipolar. That's the example. You still use the concentration gradient of sodium as an energy source to move hydrogen from low to high, but this time they move in the opposite direction. That's the antipolar. And this is the video link to the YouTube video. You can take a look. The sodium glucose importer, how to work. I'll leave this to you. Okay, the others they are reviewed because you learn them in the intro bio. How the cells make the molecule move in, move out of the cells. Vascular transport. So they create a bubble called vesicle. And depends on they move in or move out. We have the endocytosis. Endocytosis is moving in. Or exocytosis is moving the molecule out. And this process, the cell also need to use ATP. So exocytosis, example, neurons. When neurons release neurotransmitter, and they'll put those neurotransmitter in those vesicles. And when they need to release the neurotransmitter, it will cause the vesicle fusion because the vesicle is still made of phospholipid. So when they fuse with the cell membrane, and they fuse together, and this neurotransmitter just move out. So that's exocytosis. And let's look at the endocytosis. Endocytosis is moving in. You move the molecule into the cell. Could be a big molecule, could be a small molecule. So we put them in 3 different categories: phagocytosis, pinocytosis, and receptor-mediated endocytosis. And you learn that in the intro bio. Phagocytosis is sometimes called the cell eating. It's a big bite. Like the white blood cells, usually they're 2 times, 3 times bigger than the normal cell. And when they see the bacteria, they will swallow the whole bacteria in and use the lysosome, that's the organelles inside the cell. They have digestive enzyme. They're very similar to the stomach of your organ stomach. We call it organelles. And the lysosome will digest those bacteria. That's the phagocytosis. Pinocytosis, also called the cell drinking. Usually the molecule is smaller and sometimes watery, so they will take a small bite, and that's the pinocytosis. Exactly the same as phagocytosis, it's just smaller. And the third one is receptor-mediated endocytosis. Apparently, this molecule need to bind with the receptor. So some membrane protein, their receptor. When they bind with it, it will trigger the endocytosis. Pretty famous example, the HIV virus. HIV virus, it only hosts T lymphocyte. So your body have 2 kinds of lymphocyte. You have the B lymphocyte, which produce antibody, and you have T lymphocyte. T lymphocyte is the one really go to the bacteria and fight the bacteria. And the HIV virus, it will host the T lymphocyte. So they have the receptor on the T lymphocyte cell membrane. And when they bind with it, it will cause the receptor-mediated endocytosis. So now the HIV virus move in. When they move in, it's called HIV positive, so not AIDS yet. And the virus can stay in the T lymphocyte for years. That's why the HIV is dangerous, because sometimes the person did not even know they have HIV, so they can pass it to the other people. And one day when the HIV virus wake up, they will kill the host, so they will kill the T lymphocyte. And it's like, well, now your body don't have T lymphocyte to fight the bacteria. So a small bacteria, a small fungus infection can kill you. And that's AIDS. It's like your laptop does not have the antivirus software. So any antivirus can shut down your computer. So the AIDS patient does not die because of HIV, they die because of infection. Usually it's the lung infection. You can make the food very clean, you can make the water clean, you just won't be able to clean the air. So any bacteria, virus in the air, they go to your lung, and you don't have T lymphocyte to fight them, and they can cause serious infection, so they die. So we have 3 kinds of endocytosis. Okay, so that's the summary. We spent a lot of time talking about the passive transport. We talk about the simple diffusion. Osmosis, we will talk more about it next time. Osmosis is water movement. We don't call it water diffusion, we call it osmosis. Then we spend a lot of time talking about the facilitated diffusion, like the ion channels, glucose carrier, they all belong to this category. Then we talk about the active transport. The cell need to use ATP as energy source to move the molecule against the concentration gradient. And we have the primary active transport, like the sodium-potassium pump. We use this as an example. We have the secondary active transport, like the sodium glucose importer. And we also talk about you can have antiporter, like the sodium hydrogen antiporter. They don't have to move in the same direction, also belong to this category. And the last, we talk about the vascular transport, the cell membrane. They move the molecule in or out, so they all belong to the active transport category. And for the membrane-mediated transport, so this is the summary of that part. Unless the molecule is small, nonpolar molecule, like the gas molecule or steroid, they can use simple diffusion. All the other molecule, they need to use the membrane protein. And depends on what's the driving force. If the energy source is the concentration gradient, it's the passive transport. If they move through the ion channel, that's this one, channel mediated. And if the molecule is too big, like glucose, you use the glucose carrier, but it's still a passive transport. If you are moving them against the concentration gradient from low to high, you need to give them biological energy, that's the ATP, and it's called active transport. It can be the direct primary active transport or indirect, called the secondary active transport. But they all belong to the active transport category. And don't forget, when we talk about saturation, is if this membrane protein has a binding site, it can be saturated. So it can be saturated is this one and also this one. So the carrier mediated and also the active transport, all this part, they can be saturated. In the ion channel, we don't worry about saturation in normal physiological situation because they don't have a binding site, they won't be saturated. So that's where students usually get confused. So it's this and this, they can be saturated. Okay, that's it.